Here is something nobody ever told you in school. Every single detector, every rover, every instrument humanity has ever sent into space to search for signs of life was built to find one specific thing, DNA, or something close enough to it that our equipment would recognize it. Which means if alien life exists, but runs on a completely different chemistry, something our instruments were never designed to detect, we could land directly on top of it, scoop it up, run every test we have, and declare the planet dead. And here is the part that should genuinely unsettle you. NASA already knows this. They have known it for years. And they are now actively building entirely new tools to search for life they admit they currently cannot detect. Stay with me. Because what scientists have discovered about the building blocks of life in the last decade completely changes the question of what we are even looking for out there. Let's start with what DNA actually is, because most people carry a vague idea of it without ever stopping to ask a more fundamental question. DNA is not life. DNA is a solution, specific, elegant chemical solution to a very particular problem. The problem every living thing must solve is this: how do you store information about how to build yourself, copy that information reliably, and pass it on to the next generation? On Earth, 4 billion years ago, under specific temperatures, specific pressures, specific chemical conditions, evolution stumbled onto one answer to that problem. A double-stranded molecule built from 4 chemical building blocks, adenine, thymine, guanine, and cytosine, wound into the shape most people recognize. That molecule worked. It replicated. It spread. Every organism alive on Earth today, from bacteria to blue whales to human beings, uses it. But the critical thing to understand is this: the fact that DNA worked here does not mean it is the only possible answer. It means it was the answer that emerged here under these specific conditions, on this specific planet. So what happens when you change the conditions? This is exactly the question NASA decided to fund research into. And what that research produced was something that made scientists genuinely stop and rethink the entire search for extraterrestrial life. A researcher named Stephen Benner, working through the Foundation for Applied Molecular Evolution, built something called Hachimoji DNA. The name comes from the Japanese word for 8 letters, because where Earth's DNA uses for chemical building blocks, Hachimoji DNA uses 8. It stores and transmits genetic information just like natural DNA. It can support something called molecular evolution, which is the fundamental engine that drives all biological change and development. And it was published in Science magazine, one of the most respected scientific journals on the planet, with partial funding from NASA's own Astrobiology program. NASA's official response to this research was not to dismiss it. Their official statement said that scientists searching for alien life may need to completely rethink what they are looking for. That is not a footnote. That is one of the most significant admissions in the history of the search for life beyond Earth. But Hachimoji DNA is still relatively close to what we know. It uses a similar backbone, similar logic, just with a few extra pieces. The real question is whether life could run on something far more radically different. And here's where the science gets genuinely strange. In laboratories around the world, scientists have been building and testing what are called XNAs, Xeno Nucleic Acids. These are DNA-like molecules that use completely different sugar structures in their backbone. Different architecture, different chemistry, different logic. And in peer-reviewed research, scientists have demonstrated that XNA molecules are capable of both heredity and evolution, which means the two most fundamental requirements for life, the ability to pass on information and the ability to change over time, do not require DNA at all. They can be achieved through chemistry we never evolved with, chemistry that would have looked completely alien to every instrument we have designed so far. Now think about what this means for Mars, or for Europa, or for Titan. Every rover we have sent to Mars carries instruments calibrated to detect organic molecules associated with carbon-based, water-using, DNA-adjacent life. If Martian life, assuming it exists, or ever existed, solved the problem of heredity using a different molecular system entirely, our instruments would walk right past it. Stephen Benner himself recognized this so specifically that he founded an organization called the Agnostic Life-Finding Association, dedicated explicitly to searching for life on Mars in ways that would not overlook non-DNA, molecular biology. The name says everything: agnostic, meaning the search cannot start with assumptions about what life has to look like. And then there is the question of what life needs to dissolve in. Every living thing on Earth uses water as its solvent, the liquid medium in which all its chemistry happens. Water is genuinely remarkable for this purpose. It stays liquid across a wide temperature range, dissolves an enormous variety of molecules. It participates actively in many biochemical reactions. But it is not the only liquid in the universe. Scientists have studied ammonia as a potential alternative solvent, particularly relevant on moons like Titan and certain bodies in the outer solar system, where temperatures are too cold for liquid water, but ammonia remains liquid. Life using ammonia as its solvent would operate at temperatures so cold that every biological process we know would have stopped completely. Its chemistry would be radically different. Its proteins, if it had anything resembling proteins, would fold differently. Its metabolism would release different byproducts. And crucially, it would produce no biosignatures that our current detection methods were designed to find. Silicon is another element scientists keep returning to, partly because it sits directly below carbon on the periodic table and shares some of its bonding properties, and partly because it is extraordinarily abundant throughout the universe, far more common in planetary crusts than carbon. The idea of silicon-based life has a long history in science fiction. The scientific reality is more complicated and more interesting than the fictional version. The genuine problem with silicon is that its bonds with itself are significantly weaker than carbon-carbon bonds, which makes building the long, stable, complex molecular chains that life requires much harder. Silicon also reacts with oxygen to form silicon dioxide, which is a solid, essentially sand. Carbon reacts with oxygen to form carbon dioxide, which is a gas that can be exhaled, cycled, used. That difference matters enormously for how a biochemistry might function. And this is the part that often gets left out of the conversation, those limitations apply under Earth-like conditions. In extremely cold environments, in solvents like liquid nitrogen that exist on places like Titan, silicon chemistry behaves differently. Some researchers argue that in those conditions, silicon could form complex molecular structures stable enough to serve as the basis for a biochemistry radically different from anything on Earth. Not a replacement for carbon under normal conditions, but a genuine alternative in environments so cold and so chemically different that carbon-based life could not survive there at all. And then there is the arsenic story, which is one of the most dramatic and revealing episodes in modern astrobiology. In 2010, NASA held a press conference that generated enormous excitement worldwide. Researchers announced they had found bacteria in Mono Lake in California that appeared to incorporate arsenic into their DNA in place of phosphorus. Phosphorus is one of the 6 elements every known living thing on Earth uses as a fundamental building block. If this bacterium really was substituting arsenic, it would mean biology was capable of swapping out what scientists had considered an irreplaceable component. The announcement was explosive. Then 15 years of rigorous follow-up research happened. And in 2025, the paper was officially retracted after it became clear that the original conclusion was based on flawed methodology. The arsenic DNA bacterium did not exist in the way that had been claimed. But here is what that episode actually proved, separate from the specific retraction. Scientists were looking for exactly this kind of chemistry. They were seriously investigating whether a fundamental component of DNA could be replaced by a chemically similar but distinct element. They were right to look. Specific claim did not hold up. But the broader question, whether life's building blocks are truly irreplaceable or whether chemistry allows for substitution, remains wide open and is being actively researched right now. What this all points toward is something that some scientists now consider the most important reframing in the history of astrobiology. The question is not what molecule alien life uses. The question is what property life fundamentally requires. And the answer most scientists converge on is not DNA specifically. It is the ability to store information, copy that information with enough accuracy to maintain identity, and make enough errors during copying to allow change over time. DNA is one physical way to do all 3 of those things. Xeno is another, Hachimoji DNA with its 8 letters is another, and there may be molecular systems that are so chemically different from anything in our current library of chemistry that we have not yet built the concepts to describe them, let alone built instruments to detect them. This is why the scientists working on this problem now talk about what they call agnostic biosignatures. Not signs of DNA, not signs of carbon, not signs of water, signs of information processing. signs of ordered complexity that increases over time, signs of something doing chemistry in a way that random physics alone would not produce. Because if life really is fundamentally about information rather than any specific molecule, then the universe could be full of it, running on chemistry we have never seen, in conditions we have always dismissed as uninhabitable, storing and copying and evolving information in physical forms our instruments were never designed to notice. So ask yourself this. If you were an alien scientist orbiting Earth 4 billion years ago, carrying instruments designed to detect the specific biochemistry of your own world, would you have found us? Or would you have pointed your detectors at our oceans, seen nothing that matched your expectations, and moved on, confident that this particular planet was empty? The question of what aliens are made of is not just about them. It is a mirror pointed directly at every assumption we have ever made about what life is allowed to be.